Now we discuss about class 12th biology, chapter 6 molecular basis of inheritance. First of all, we discuss about what is DNA. DNA, that is full name is deoxyribose nucleic acid, is the molecule responsible for carrying genetic information in all living organisms. It is a long chain made of deoxyribonucleotide and its length is determined by number of nucleotide base pair. This is the structure of DNA. We see the double helix A, T, G, C, the sugar phosphate backbone, nitrogenous base. Watson and Crick were the first scientists to propose the double helix model of DNA. Using X-ray crystallography, each strand of DNA is polymer of nucleotides. Each nucleotide consists of three components: a deoxyribose nitrogenous base, and phosphate group. DNA follows the central dogma of molecular biology, where the genetic information follows the DNA to RNA and then protein. The structure of DNA resembles twisted ladder. Two strands are held together by weak hydrogen bonds between paired nitrogenous base. In this pairing, purine bases adenine or guanine always pairs with pyrimidine bases thymine and cytosine. Specifically, adenine, that is A, pairs with thymine T and guanine G pairs with cytosine C. Structure of polynucleotide, nucleotide, phosphate group. First is phosphate group, that is P and double bonded O and OH hydroxyl group, and the nitrogenous, second point is nitrogenous base, and third is pentose sugars. This is the nucleotide bases. Structure of polynucleotides. Polynucleotides is a long chain made up of repeating units of nucleotides. Polynucleotides made up of repeated unit of nucleotides, which are the building blocks of DNA and RNA. Nucleotide structure. Each nucleotide consists of three components. Nitrogenous base is classified into two: purines and pyrimidines. Purines that is adenine A and guanine G. Pyrimidine, which is cytosine C, thymine T in DNA and uracil in RNA. Sugar. The sugar is pentose, which between the DNA and RNA. The deoxyribose in DNA and ribose in RNA. Phosphate group. The phosphate group connects nucleotides together by linking the sugars of one nucleotide to the phosphate of the next, forming the sugar phosphate backbone of the polynucleotide chain. The combination of sugar and nitrogenous base form a nucleosides, while a nucleosides with added phosphate groups becomes a nucleotide. These nucleotides link together to form a polynucleotide chain that makes DNA or RNA. Now we discuss about gene. What is gene? A gene is a functional unit of inheritance responsible for transmitting genetic information from one generation to the next generation. In eukaryotic organism, DNA is composed of both coding and non-coding sequence of nucleotides. The coding sequence are called exons, and the parts that codes for protein. The non-coding sequence is introns, do not code for proteins. Coding sequence exons, non-coding introns, which is not codes for proteins. While exons are included the mature RNA after the processing, introns are removed. Structure of RNA ribonucleotides, that is thymine, cytosine, adenine, guanine. It's RNA. RNA is a single stranded nucleic acid found in all living cells. It acts as a messenger, carrying instruction from DNA to help control the process of photosynthesis. There are three main types of RNA. First is mRNA, which is messenger RNA. which carries genetic codes from DNA to ribosomes where the protein are synthesized, and second is transfer RNA, that is tRNA, brings correct amino acid to ribosomes during the protein synthesis, and third is RNA, which is ribosomal RNA, combines with protein to form ribosomes. It sites for protein synthesis. Next is packaging of DNA helix. How packaging? In prokaryotes, DNA is organized as a large loop in the nuclear region. Here, the negatively charged DNA is tightly held to positively charged protein. In contrast, eukaryotic DNA is arranged in the more complex structure of chromosomes. In eukaryotes, DNA is wrapped around the core of histone proteins to form a structure, nucleosomes, and the nucleosomes consist of DNA wound around the histone octamers, which is made up of 8 histone proteins. These histones are rich in basic amino acids like lysines, arginines, gives positive charges. There are 5 types of histone proteins: H1, H2A, H2B, H3, and H4, with the histone octamer containing 2 molecules, each of H2A, H2B, and H3 and H4. This packaging helps to regulate the gene expressions. Nucleosomes prevent the DNA from becoming tangled and contains about 200 base pairs, that is BP, of DNA. The further organization of chromosomes, the further organization of chromatin, which is DNA plus protein complex, is added by non-histone chromosomal proteins, which is NHC. Next, euchromatin. There are transcriptionally active regions of the DNA where chromatin is loosed, packed, and appears slightly stained. Heterochromatin. There are the transcriptionally inactive regions where the chromatin is densely packed and taking a darker stains. Next, RNA worlds. RNA is believed to have first genetic materials with substantial evidences sustaining the essential life process evolved around RNA. It several dual rules acting both the genetic materials and the catalyst. However, RNA have highly reactive nature made in unstable catalyst. This instability leads to the information of DNA from RNA with chemical modification that is enhance the stability. Now we know about replication. What is replication? Watson and Crick proposed the DNA replication, which is semi-conservative, meaning that each new DNA consists of one original strands and one newly synthesized strands. This was experimentally confirmed by Nielsen and Stahill in 1958. Taylor et al. Conducted experiment using the radioactive thymine is the fava beans, Vika fava, to further demonstrate the DNA replication is indeed semi-conservative. The enzyme DNA polymerase plays a crucial role in DNA replications. Catalyzes the process. It can only two nucleotides in 5 strands to 3 strands directions. This results in 2 different replication process. First is the leading strand, second is lagging strands. Leading strands replication continue along at these strands, where the template starts from the region of 3 strand to the 5 strand. And lagging strands replication discontinuous. Yes, because the template runs from 5 strands to the 3 strands. This strand is synthesized in short segments known as Okazaki fragments that are the latter joined together. Next is transcription. Transcription is the process through the genetic information DNA is copied into RNA and specifically a focus on segment of the DNA at a time. During the process, adenine pairs with uracil instead of thymine in DNA. Transcription involves 3 regions: a structural gene, promoter, and terminator. The enzyme RNA polymerase catalyzes the transcription process and the direction of transcription mirror that DNA replication processing from 5 direction to the 3 direction. And the strands DNA that serves as a template of RNA synthesis is called the antisense strand, which has 3' to 5' strands. The coding strands contain the structural gene along the promoter and terminator region and includes the both exons, which is coding sequence, and introns, which is non-coding sequences. Now we discuss about genetic codes. The genetic codes refer the sequence basis of the messenger RNA, which is mRNA, that specify the order of amino acid into protein synthesis. Each code consists of 3 nucleotides forming the known as codons or triplets. There are total 64 possible codons. 61 of codons correspond to the specific amino acid, while remaining 3 are the stop codons because they do not have for any amino acids. The codon AUG serves as a both start codon and codes for the amino acid methionine and making the beginning of protein synthesis. These codes for the essential for translating the genetic information for mRNA to functional proteins, which are vital for the structure of the functions of the cells. Now, mutation and genetic code. Mutation involves changes in genetic sequence, and one common types of point mutation where the single base pair is altered. A known example, sickle cell anemia, caused by the mutation in gene code for the beta globin chain for hemoglobin. In this case, amino acid glutamate is replaced by valine, leading the seeking of red blood cells. Another type of mutation is frame shift mutation, which occurs when there is a loss or gain of one or two base pair in the DNA sequence. These changes leading frame the point of mutation, altered the entire sequences of the amino acid downstream. Frame shift mutation can have significant impact on the genetic protein, often leading the non-functional proteins or several genetic disorders. Next topic is translation. What is translation? Translation is the process through the amino acid linked together from the protein, translated by the peptide bonds. During the process, all types of the RNA messenger, RNA, mRNA, transfer RNA, and ribosomal RNA played role.